right, I hope everybody can hear me. The guy who's supposed to go before me, he'll go a little bit later. He's right over there, for those who are interested. Okay, so to start off, I do have just one slide, a couple slides of advertisement, because I work for a small biotech that probably not many people have heard of, Profectus BioScience. This sort of describes who we are. We do clinical stage work with vaccines. Our technology revolves around using a negative strand RNA virus, VSV, vesicular stomatitis virus. So we have a nice little phrase, vesiculovax, for those vaccines based in VSV. We also use a prime boost regimen using DNA with electroporation, and we've heard a lot about that in the recent talks a few last few days. And we prefer to use the device developed by ICOR. They're a longtime collaborator. And we also do subunit-based vaccines as part of our core technology. Our pipeline has a number of different approaches going on, including in biodefense, as well as chronic infectious diseases such as HPV, HCV. And we're doing prophylactic vaccines against HIV, and that's the one I'm going to focus on here today. All right, so for HIV, obviously the start, as a company guy, we kind of try to figure out what we want to achieve out of any vaccine and in our target product profile. And this is based on the current thinking for HIV. We can always debate this, but we have obviously want a minimum of 70% efficacy, and these are from mathematical models that suggest that at that level we can have a pretty good impact on the epidemic. How to get there? Well, that's a good debate. Broadly, we would say broadly reactive antibodies that prompt some sort of antiviral effector function, ADCC, neutralization, CD8 T cells that kill, we think is a key part, and help our population. And this is some newer observations that have come out recently where these CD4 T cells are able to produce antiviral chemokines that block or prevent the virus from infecting, so you don't lose your CD4 cells. So this is kind of something we're looking for. Currently, most of the vaccines that we have for HIV are administered IM. Ideally, you'd like to get everybody responding to these things once you get them in. Currently, the vaccines, usually 2 or 3 or more immunizations are required. Boosters, big question. Longevity for HIV is a big issue. And protection that can last for much longer than 1 year. As may remember, the famous RB144 study had early protection, upwards to 60%, that waned quite dramatically over time to near 30% at the end. Okay. The approach that we're taking for HIV vaccines we call full-length single-chain, FLSC, and it's a chimeric protein, subunit protein, consisting of GP120 genetically fused to a fragment of CD4. And basically what we're attempting to do here is mimic one of the early steps of HIV infection, that being the point in time when GP120 engages CD4. This construct wraps upon itself. The CD4 binds to GP120 and it causes a series of conformational changes in the protein, as you would predict when HIV joins its target cell. Now, how does it work? How do we think it might work? Well, it's based on the premise that chronic viruses tend to hide their vulnerable parts. And so the basic premise is by altering the envelope structure using this envelope receptor complex, we can then change how the antigen is presented to the immune system and enhance the immunogenicity of potentially conserved, maybe vulnerable parts, or expand the epitope diversity in the antibody response. Now I want to contrast this with the approach that my colleague Rick Wyeth has presented earlier, where they're trying to replicate the full-fledged trimer in a soluble subunit. This approach is a little bit different from that. So, okay. Now, does single chain protect? So we've done a number of experiments in rhesus macaques in a number of different models, and we've come to the conclusion that it can protect. and it does seem to be related to the ability of the immunogen to induce what we call CD4I response, or a response directed towards the CD4-induced epitopes, that these responses have to be above a threshold. There appears to be this ADCC activity that's important, but we cannot have an overabundance of T cells, and then we coin the phrase immune balance. So I'm going to go present some of the data that has led us to this conclusion. The first study we did was one in macaques, where we had 6 animals per group, and we're looking at single chain or GP120 formulated in either a couple different adjuvants here. The single chain in this case is RH FLSC, and that RH stands for rhesus, single chain, rhesus CD4, and a rhesus macaque, homologous, so we're not getting an anti-CD4 response. Animals immunized, the schedule here, and then at the end we're challenged multiple low doses with SHIV 162P3, which is known to be a CCR5 using SHIV. It's neutralizing resistant. Now, the important thing to remember here is that we decided to take a very hopeful and optimistic approach and powered the experiment to look for greater than 82% protection, the slam dunk approach. So if we got it, we win right out of the box. And as you could probably imagine, that was probably a little too hopeful, but we learned something from it. All right, so here is basically the Kaplan-Meier plot for all the groups. And as you can probably see, there's nothing really that jumps out at you except perhaps one group that received single chain in an adjuvant RC529. And there was a modest improvement of protection in this group. All the animals that were immunized with single chain had an anti-CD4I response that were directed, of course, to antibodies 17B, A32, 19E. These are all antibodies that bind to these CD4I antibody epitopes. I just wanted to show you that indeed the antigen did what it was supposed to do. And the animals that had the higher anti-CD, we took all the animals and did a meta-analysis, post hoc meta-analysis. We found that the animals which had the higher CD4I titer, is indicated by this this particular antibody, N12I2. It's a newer antibody against the CD4I epitope. Those animals appeared to have better protection than those that had a less than a median titer. Now, this did not track with antibodies to the CD4 binding domain, as you can see over here. Now, I remember I said the key is immune balance. So one of the other things we noticed is that in certain groups, and in particular those animals that received ISCO matrix, there was a very significant T cell response. So we were curious, is that obviously that HIV infects T cells, CD4 positive T cells, whether or not we actually improved or made the protection worse by making more targets. So we asked that question in this way. Taking ELI spots and when dividing the ELI spots in IL-2 ELI spots above and below the mean, we noticed that animals which had very high IL-2 ELI spots became infected rather rapidly, while the animals which had lower ELI spots below the mean seemed to be better protected. Now I want to make one small note about the statistics here. This is a Gershorn statistic which weights early protection over long-term protection. So as you can see, there seems to be an early protective response here. All right, now, so since there seems to be this relationship, we ask the question, well, if we remove those animals which had this high T cell response, now do we see some correlates? And indeed we do. So the correlation in those animals which did not have high T cell response correlated with single chain titer, ADCC activity, and competitive titer against this one particular epitope. Now, this is a CD4I antibody epitope, and it's towards the C terminus of GP120, and it's thought to be important for protection that appeared in the RV144 study. So that's kind of convenient. All right, so when we graph this using ADCC responses to ELI spots, in this case gamma interferon, they matched up pretty well with the IL-2. we see this, what appears to be this peak, what we call a zone of immune balance, where if you get the right amount of ADCC responses with the right amount of supportive T cells, you can get a better protection. So we decided to continue to test this idea in a follow-on experiment, which we had, where we had animals, again, immunized with single chain. This time we had another group immunized with single chain with TAT toxoid, and I'm not going to belabor it, but it's a soluble subunit-based antigen that there was some interest in with this particular molecule in our group. We formulated in an adjuvant, GPIO 100. It's a saponin-derived adjuvant like QS21. Unfortunately, we didn't have available stock of the RC529 with us, but we knew from preliminary experiments that the amount of immune response we generate with single chain was comparable when we used this formulation of GPIO 100 with this TLR agonist with RC529. So similar schedule. In this kind, we're using a multiple low-dose challenge, and because of how the challenges turned out and the level of protection we achieved, over time we had to give increasing amounts of the virus. And this is just some of the different differences between the first study and the second, which I just went through. Okay, so here's the basic results. We can see that in the animals that were controlled, just receiving adjuvant alone, that had this profile. The animals that received the TAT toxoid seemed to be very close to the adjuvant control, while the animals that had single chain alone were much better protected, so giving us a log rank of 0.03. So what was interesting, the TAT toxoid seemed to enhance T cell responses over what we saw with the single chain. ADCC responses in terms of plateau cytotoxicity were higher in those animals that received just single chain alone. And so generating we wanted to say an immune quotient of immune balance, ADCC over gamma interferon, where the animals that received just single chain seem to be much better than the animals that received toxoid. We also noticed through this process is that TAT toxoid, for those of you who may know, is a protein that can help, many people use to help get other proteins inside cells. And it does this through an association with the target protein. And in this case, we noticed that TAT toxoid also associates with single chain. And so we believe that what happened is that TAT toxoid actually enhanced T cell responses by causing more of the single chain to get into the cells. Okay. Third study. Now this time we decided to switch to a different challenge system. Instead of using SHIV, we did SIV, and these are two prevailing models. You can debate which one is more relevant. In this case, we're using a DNA prime using a single chain derived from SIV, in this case E660, and have 5 different groups with 8 animals per group. And again, this is going to be powered to be about 75% efficacy. Now the key difference here is that we had animals that were given different adjuvants, genetic adjuvants. In one case, LTA1, heat labile toxin A subunit, recombinant IL-12, rhesus IL-12, and then the combination of the two. And these are given as DNA and injected through electroporation. At sometime after the 3 DNA immunizations, we also gave a subunit boost of single chain in a derived from a transmitted founder version of SIV, and this was given in aluminum hydroxide. The challenge important here, very important, is a 251 challenge, which is completely heterologous to E660, and the genetic difference between these two isolates and this is the same difference as would be in a clade, so from like clade B to clade C. Okay. Again, here are some of the key differences that I just went through. All right, so why do we use IL-12? Well, there was some data out there that IL-12 does enhance NK cell activity and is known to enhance ADCC is well-established Th1 adjuvant. I'm sure everybody knows that. So we thought, well, this could be a good way to amp up our ADCC responses. LTA1 is from heat labile toxin of E. coli. It's given just as the A1 subunit, devoid of the B subunit, and it's given through DNA. It activates adenylate cyclase, and then this causes an immune activation. We know this is also in our mouse studies. It does a really good job of amplifying T cells, and it works together with IL-12. Now, so we thought, well, maybe we can get an opportunity to get a better ADCC response by combining these two. Now, with the caveat that we may actually get more T cells as well. So here's our genetic adjuvant based on, sorry, this should be LT here, LT instead of CT, but they're the same. This is what our construct looks like. Additive, effective, LT and IL-12. Okay, all right, so challenge data. Inclusion of IL-12 seemed to give us superior protection. And as you can see, this red bar here, this is a Kaplan-Meier of all groups. And this is here with the IL-12 group isolated against the others. So the IL-12 seemed to be giving us some magic here. IL-12 was better than DNA by itself, suggesting that inclusion of this adjuvant is a key aspect of the protection. And when we added in more controls that were donated to us by collaborators from other experiments, we got quite a significant level of efficacy. These were historical controls from naive macaques that received the exact same challenge virus at the exact same dose, administered by the exact same technical staff in the exact same animal facility, all done within about 6 months to 1 year of our study. So these historical controls we feel are were good ones to pick. They're not just random historical controls. All right, so what's helping us here? Okay, well, IL-12 and IL-2 combination, like we saw in our mouse studies, did enhance T cell responses, and this is looking at ELI spots over the entire period of immunization. We could see that there was a significant increase in T cell responses in the IL-12 and the LTA1 group. And IL-12 also by itself enhanced ADCC, as we can see here. And interestingly enough, we could also see an increase in the groups that received IL-12 in antibody binding to Fc receptor 3A over R1. So 3A is a ADCC receptor. R1 is a general binding receptor for antibodies. Okay, so these two things, this tracks. So again, going back to immune balance. So now if we parse these animals into quartiles, where we say, okay, let's look at the animals which were ADCC negative versus those which were ADCC positive but had interferon gamma responses less than the 75th percentile, and that was about 15 animals, versus animals which were ADCC positive and had very high T cell responses in the 75th percentile or above, 8 animals. There was a significant level of protection with the middle group, that's the red here, suggesting that you got to get the right balance of ADCC without an overabundance of T cells that potentially could complicate things. Okay, so this was very encouraging to us, and all along we've been developing this product as a potential vaccine for use in humans. So here's a quick summary of where we stand in that effort. We have drug substance manufactured from a 293 cell line that is currently been manufactured and released with our about 1 gram per liter final yield. Alum formulation, 95% absorption, been stable for about a month at 37, and we're still going. We have already had discussions with the FDA. Standard rabbit tox is actually nearly done now. We have immunotox studies that the FDA requested. And the caveat there is that because this thing has CD4 in it, there's always a concern that we may induce an auto anti-CD4 response that could be deleterious. So we're doing a fairly significant NHP study to address this concern. Single chain has been in over 200 macaques so far without any evidence of this kind of response, but it's important to look for it. We have about 3,400 vials of the drug product available for testing and more unformulated for other trials. And as I said, there was an immunotox study. This is sort of the general design. I won't go through much detail, but basically what we're looking for is any kind of deleterious auto-CD4 response in macaques. This includes drop in CD4 count, impact to immunogenicity of a second T-cell dependent antigen, KLH, in this case, or inhibition in a mixed lymphocyte reaction. This is a very extensive study and very expensive, as you can imagine. But we think this will answer the question fairly easily, whether or not we will get any of these problems. Now, if we do observe any kind of, again, deleterious is the key here. Just any CD4 response that's just there doesn't mean it's going to be a bad thing. We're looking for changes that would potentially cause a problem. If so, then these additional safety measures will have to be added to the clinical protocol. And we expect to be in the clinic about mid-2015. So our goal is to improve our potency and durability with the right, one more slide, SC-mediated protection, right balance of supportive T cells, and we hope to achieve that through a number of different clinical trials looking at either DNA priming, new adjuvants, or potentially incorporating our VSV strategy. And here's all the folks that have been helpful. And important to note, funding for this program has been through the Bill and Melinda Gates Foundation, of course, in NIAID, and the Henry Jackson Foundation. So that's it. Thank you.
We'll take 2 questions from the audience. Are there questions? Yes.
I'm interested in your use of TAT as an immunogen. I wondered, given the effect it had, if you'd consider still using TAT, but at on a different schedule to your other immunogen.
Yeah, that's a good point. You potentially could use TAT if you didn't incorporate it with another protein antigen. There is some evidence that anti-TAT antibodies may be potentially useful. But in this case, what our objective is to try and simplify the regimen as much as we could. So that's why we try to say, well, let's just take it out.
Yeah, Tim. So you think it's this balance you need to strike? So you think it's just strictly too many CD4 positive target cells is why you're getting
that's our
protection?
hypothesis. Yes. So we've done a lot of polyfunctional flow as much as we can in these studies. The animals that were protected, a lot of times we can detect animal responses, T cell responses by ELISpot, but we really can't by polyfunctional flow, and that's just a reflection of the sensitivity of the assays. There was a nice presentation by CTL yesterday about the differences in ELISpot and how sensitive it is versus flow. So we don't yet know what are the culprit T cells. We suspect they're probably Th17 because LTA1 is really good at inducing Th17. So that's what we're going to look for and see if that's our key target. Our bad boys in our mix.